Good evening, everyone, and welcome to Strait Science. Strait Science is put on by the University of Alaska Fairbanks, UAF Northwest Campus in Nome, and also UAF Alaska Sea Grant, also in Nome. You're in the Nome office tonight. And UAF Northwest Campus and Alaska Sea Grant are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. Now, tonight, we've got Jim Murphy, and Jim Murphy is a research biologist with the NOAA, N-O-A-A, Fisheries, Alaska Fisheries, and he is coming to us tonight live from the Auk Bay Labs of the Alaska Fisheries Science Center in Juneau. Oftentimes you may hear about NOAA Alaska Fisheries Science Center, but you always have to ask, are you out of Seattle or are you out of Juneau? And actually Jim is an Alaskan and on our time zone and giving it to us straight up. So we appreciate it, Jim. The work you're doing from Juneau and for the rest of the state, and that you're Alaskan and in our time zone. And tonight, Jim's going to talk about the 2023. He's got ecosystem and surface trawl survey. So I know it involves a little bit of a surface trawl, but he's going to talk about all the other types of equipment they use to get at this. And this is, we heard earlier this year about the bottom trawl survey and what they found with the pollock and cod and the king crab and things like that, Arctic cod and tom cod. But we're going to hear more, I think, and Jim, I'll hand it to you in a sec, but we're going to hear more about salmon, young-sized crab, things like that. So this will be a good partner to the previous Strait Science we've had from the bottom. So with that, thank you so much for being here, Jim Murphy, and take it away.
Well, thank you, Gay. Yeah, I think our surveys kind of start from the top and work down, whereas the bottom trawl surveys are working from the bottom on up. But so let me just first of all, I want to point out that there are a lot of different people that are involved with this survey, a lot of different agencies, and these surveys are quite challenging to pull off. We couldn't do it without the support of many different agencies and scientists. So let me see if this is working. There we are. So the survey, the figure here shows the stations that we typically sample, although they've varied from year to year. But it was originally set up at the beginning to be a focus study on juvenile salmon and trying to gain a better understanding of their marine ecology. And since then, we've expanded the survey a bit, trying to address the broader ecosystem, and that's brought in many more scientists and helps us understand more completely the connection that salmon have with the Northern Bering Sea ecosystem. So let's see if I get this right. So I'm having trouble. There we go. Okay, I'll use this. So with concerns over the warming temperatures in the Bering Sea, obviously water temperatures, and in this case, we're using sea surface temperature, is one of the key products that we produce from the survey. There's a lot that's kind of going on with this figure, but the blue lines and the blue dots are the average temperatures that we measure with the CTD. And the gray line is kind of a reconstruction of temperature with satellite data. The main point here is that temperatures in 2023 were actually dropped to slightly below average. So we're seeing a fairly abrupt cooling of the Northern Bering Sea from the warm temperatures we saw in 2019. So a lot of the, and then the figures on the right show, the top figure shows the sea surface temperature. So you can see sea surface temperatures are always warmer up along the coast. And then the spatial map on the bottom right is the actual temperatures, bottom temperatures. And here you can see the presence of this cold return of the cold pool. And so that has had a major effect on the cooling of the Northern Bering Sea. So one of the things that we measure in addition to temperature in the physical oceanographies, we measure zooplankton abundance. The figure in the center, in the top, is the abundance index for large copepods. So these are copepod species that are well known to carry a lot of fat. So it's always, this particular prey group has been very important for understanding the transfer of lipids and stuff to higher trophic levels. So what we saw was in 2019, we saw a record low abundance of these large copepods. But we've started to see them increase in recent years, and this is likely just the cooler temperatures that we're seeing. We're seeing large cope abundance being closer to average. But we're also seeing, so that what's the very opposite pattern is true for small copepods. So we saw the record abundance levels of small copepods in 2019, the very warm year 2019, and we've seen their numbers drop to almost record low abundance levels in 2023. So we began sampling, began a benthic sampling program in 2021, and so we're just now starting to get a better understanding of how the benthic community is changing over time. But the figure on the left shows the size distribution of juvenile crabs. So these are going to be very small crab, like what we consider to be the smallest cohort would be less than 15 millimeters, so kind of the width of your pinky. So these are very small crab. But the interesting result here is you can see the progression of that cohort through time. So we can actually track that there was a relatively good recruitment in 2021, and that has progressed over time over the last 3 years. But we are seeing almost no juvenile crab in that very small size category in 2023, which is of concern. On the other figures here, you can see that the young of the year Pacific cod was higher. There's lots of variation in those estimates, but it's higher. That means that I think the cod are doing well in the Northern Bering Sea. But the real big change was the large increase in the young of the year saffron cod. So these would be identified as the tom cod. So we're seeing kind of a return of these saffron cods to the Northern Bering Sea, which is a positive sign. So as part of our surface trawl operations, we'll monitor forage fish. And so these are just two key forage fish species. There's Pacific herring on the left and capelin on the right. And we typically see higher biomass of herring and capelin in the Northern Bering Sea compared to the Southern Bering Sea. But we saw both species drop to kind of the lowest levels that we've seen in the entire history of this sampling program during 2019, that really warm year. We're seeing some evidence of a recovery of the abundance of these species, but their abundance still is not, they haven't returned to the levels that we saw in the previous cold years. And so obviously temperature will have a very direct and immediate effect on the abundance of these species and their distribution. But temperature effects on survival will take some time before these species will recover to kind of cold conditions. And
Jim, this is Gay. I'm just going to say, for my brain, I just want to reiterate, on the left is herring and the right is the capelin. The black dots are the Northern Bering Sea numbers, right? And the lighter blue dots are from the south
Southern
of
Bering.
like St. Matthew, sort of like south of St. Matthew.
South of Nunivak Island.
Okay, thank you.
Yeah, sorry, I should have made that a little bit clearer. But yeah, it's just I think it's concerning to see the large decline in forage fish species in the Northern Bering Sea, and this is there are a lot of predator species that are dependent on these forage fish, Chinook salmon being one of them. but a lot of the seals also rely on these forage fish species as well. So there's we'll see. We're hopefully it's good to see some recovery of these species, but it would be nice to see their abundance levels increase quicker. Hopefully, we'll see some improvement soon. Kind of related to this topic to a certain extent is the research that we started in 2021 on monitoring Ichthyophonus. So this is kind of a busy table, but so we have 3 years of data. The first column shows the infection rates of juvenile Chinook. So as you can see, Ichthyophonus is not present in any of the juvenile Chinook.
But what is Ichthyophonus? Sorry.
Ichthyophonus is a parasite that Chinook salmon get infected with. It infects their heart and their major organs, and it's a major source of concern in the Yukon River, and it's a major concern for the mortality that's occurring in Yukon River Chinook. So moving to the next column where we have immature Chinook. So what we can see is that immature Chinook salmon are infected with Ichthyophonus. And so this occurs throughout their immature life history stage. And that varies from year to year. Both the prevalence of Ichthyophonus as well as the intensity of Ichthyophonus varies in the immature population. But we did see a reduction. We're seeing a lower prevalence and a reduced intensity of Ichthyophonus in the immature life history stage. So that's a positive sign for Chinook. And no herring are detected. Ichthyophonus is not detected in herring, rainbow smelt, sand lance. Very small levels of Ichthyophonus in capelin, and no pollock had Ichthyophonus. But where we saw the most likely source of Ichthyophonus in Chinook salmon was in the presence of age 1+, which is in 2023. Like 33% of all the pollock, this would be age 1. It was pretty much all just age 1 pollock in 2023, but 33% of them have Ichthyophonus. So this is likely the source for Ichthyophonus in Yukon River Chinook salmon. So this is kind of this describes the diets of juvenile Chinook on the left and juvenile chum salmon on the right. It's busy, but the two bottom rows for Chinook are capelin and sand lance. So you can see that Chinook salmon tend to feed very heavily on fish. They're very piscivorous. But in recent years, since this warm period, you've started to see a lot more diversity in the diets of juvenile Chinook salmon. And the figure on the right, the most common prey item for juvenile chum is gelatinous zooplankton prey. And you can see that is fairly consistent. It's variable from year to year. Overall, the diet of chum salmon is much more diverse than Chinook. But we're also seeing a change in the foraging patterns and the diversity of the diet of juvenile chum as well. This figure here shows how temperature is altering the energy density, which is really follows the lipid content, so it's their condition. So temperature has this sort of dome-shaped effect on the condition of juvenile salmon. And it's much more pronounced in species that are planktivorous, like chum and pink. So you can see these really warm years, 2019, energy density of all species of salmon were quite low. And Chinook salmon, there is a dome-shaped pattern there, but they tend to be more stable. And I think a lot of it has to do with the fact that they feed on fish. So they don't have as much variation in the quality of their diet as some of the other species do. But probably the most important takeaway message in these figures is that in 2023, we saw record high condition for chum and pink. And so we're seeing the juvenile chum and pink salmon are in the best condition that we've seen them for the last 12, 13 years. So the other aspect that we have been monitoring and studying in the Northern Bering Sea is just the overall nutritional quality of the diet for Chinook salmon. So it's not just looking at the amount of lipid or the quality of their prey. We have to look at other factors such as the amount of thiamine or vitamin B, because Chinook salmon, we're seeing evidence of thiamine deficiency of Chinook salmon in the Yukon River. So understanding the amount of thiamine that are in the prey species that they eat is important. But it's also important that we understand the amount of thiaminase, which this is an enzyme that breaks down thiamine or vitamin B. And so if Chinook salmon are feeding on prey that have a lot of thiaminase, they're going to become thiamine deficient. So this is just, again, following through. It's rainbow smelt is actually at this point is the only species we've seen that has significant levels of thiaminase. So this is probably where and how Chinook salmon are becoming thiamine deficient, is by feeding on rainbow smelt. So I'm going to talk a little bit more about the abundance of Chinook salmon. So this is the cooperative work we do with Fish and Game as part of the Alaska Sustainable Salmon Fund. This figure here shows the mixed stock abundance of juvenile Chinook. So this is all juvenile Chinook stocks in the Northern Bering Sea. The figure on the right just shows the distribution in 2023. So here we can see this overall declining trend in the abundance of juvenile Chinook in the Northern Bering Sea. We also monitor the genetic stock composition of Chinook. So that allows us to identify the stock-specific abundance, which gives us much clearer connections to the adult returns and how it affects and being able to interpret survival. So in 2023, although we saw the U.S. stocks within the Yukon River, this would be the lower Yukon and middle Yukon, and the middle Yukon, both of these stocks had higher than average proportions in the Northern Bering Sea. But I think a lot of that has to do with the fact that the upper Yukon, or the Canadian origin stock group, was well below average in 2023. And so that's a little concerning, particularly for the Canadian origin stock group. So these figures are just that, very similar to the mixed stock abundance, but it's actually for specific stocks. Canadian origin, which is the upper Yukon, and then just the total all Yukon stocks. So those are the two stock-specific abundance indices that we provide for the Northern Bering Sea. The figures in the center show the relationship between juvenile and adult abundance. So that kind of gives us a way to look at what the variation is in marine survival after this juvenile period. And then the two figures on the right are actually the run size forecasts for those two stock groups. And one thing that we've noticed is that for the Canadian origin stock group, the actual run sizes have been coming in well below the forecasted run sizes for about the last 6 years. So we're seeing this sort of departure from expected we're seeing the run sizes just not reaching the expected run sizes based on juvenile abundance. And some of this is, and the difference in, excuse me, I'd say if you look at the total Yukon forecast, we're not seeing that same pattern. So we're seeing more consistency in the total Yukon forecast. So this could reflect a combination of the mortality of these two stock groups of salmon, but it's probably more tied to how there's in-river mortality as these upper Yukon fish are migrating up the river, they're not surviving. So that will generate a divergence in our forecast models for the Canadian origin stock group. But there's other aspects, like these two stock groups have different approaches in how the run size are estimated in the Yukon River. So we still have some work to figure out what is going on and why the Canadian origin forecasts are not lining up with the juvenile based forecasts. So let's see, the last figure I have here is this work that we're doing on the fall Yukon River chum salmon stock group. So these are primarily middle and upper Yukon stocks of chum salmon. And this is the abundance index that Sabrina Garcia has developed from the for this stock group in the Northern Bering Sea. And the models are still in development. And they're a little more complicated than the Chinook model because we have to correct for changes in marine survival for chum salmon to provide a meaningful forecast. So we're working through that. Also, the Yukon chum salmon, at least the fall Yukon chum salmon, are also present in the Southern Bering Sea. So it takes a full Eastern Bering Sea survey data to adequately measure their abundance over time. Let's see. So just to help, I want to emphasize again, just like this survey is not possible with a lot of different groups and many different scientists. Kind of one of the key sources of funding for the survey is the Alaska Sustainable Salmon Fund, and this fund is these proposals and this research project is collaborative with Fish and Game. So it's really focused, that's where the focus on juvenile salmon abundance and run size forecast models are supported through AKSSF. The benthic and zooplankton ecology work is supported by EcoFoci in Seattle. So Dan Cooper, Dave Kimmel, and others have been integral to get this component going for the survey. There's a lot of folks within the recruitment, energetics, and coastal assessment program here at Auk Bay Lab in their laboratory support for trophic ecology. And then there are several scientists within the Pacific Marine Environmental Lab and UAF that are helping us with the oceanography and phytoplankton ecology. And finally, Liz Lubunsky, who I just saw, she's here, but she's with the U.S. Fish and Wildlife Service and has been helping to monitor seabirds since about 2007. So all these people come together on the vessel, and we do our best to keep all these indicators and going every year. So I think with that, I will open it up to questions.
All right. All right. Well, thank you. Thank you, Jim. I've got a lot of questions and I'm sure others might have. Thank you for such a nice presentation. And while people are thinking of their questions, and you might want to hit stop share screen so people can get a look at you, Jim.
Okay.
Yeah.
Well, I want to leave this. Okay, I'll do that. Can they see me? I'd like to leave it up in case people have questions so I can.
Oh, I do have questions on the slide, so maybe other people will. But for the Straight Science audience, while I see already there's a number of hands up and stuff, so that's awesome. We will, this is a time to thank Jim in the chat. Show a little love for the Straight Science speaker. It's never easy, regardless if your message is good news or bad news, it's never easy being a Straight Science, any kind of public speaker. So we're a good audience out here in the Bering Strait region, and we appreciate when people tell us, take the time to give us some results from the research community. So drop something in the chat if you feel like it. And I see starting off, we've got Dean Stockwell and Franz. So go ahead, Dean.
First of all, nice talk. Liked it a lot. I have a question for you from the beginning of your talk. How do you separate temperature effects from predation changes having their effects on the forage fish? For example, when the cold pool diminished, you had a lot of large adult pollock move north. How do you separate that from what you're seeing in temperature as an effect on whatever species you're looking at?
Oh, that's a good question.
Sorry.
Yeah, no. That's a good question because what we see is what we've seen before is that and it still could be an interaction there, but we typically see, particularly for capelin, is that as it gets warmer, it's kind of like their distribution contracts north. So they tend to be very temperature sensitive. Now, it's also possible that, like you're saying, that part of that contraction of their distribution could be predation pressure. And I think that's an excellent point. And yeah, no, that's a really excellent point. I think for me, it was really shocking to see the large decline in herring. And I think it's not just a distribution shift, because if it's distribution shift, we would expect herring abundance to be back up to the way it's always been, because temperatures are actually below average, but we don't see that response. So I think it's a survival. So that could be predation, that could be other factors as well. But I think it's a survival signal.
Thank you.
Thank you.
All right. Thanks. Thank you, Jim. Franz, go ahead.
Hey, Jim. Whoops. Not sure if you can see me, but apologize. I'm also cooking at the same time. But thanks, Jim. Great talk.
Okay.
I was actually wondering about some of the gadgets that you showed earlier, and just to make sure that I'm clear on the cod and saffron cod plots that you showed. Yeah, right there. So you only have the last 3 years here, and that's because that's when you start sampling in 21, right? And there isn't really a baseline for any of these, because you said something about saffron cod and maybe Pacific cod recovering, but you don't really have anything to compare that to for earlier years, right, from the Northern Bering Sea.
Yeah, I can help explain that. We actually catch the Pacific cod and saffron cod young of the year in our surface trawl surveys. So we have those time series that go all the way back from the beginning.
Yeah.
So what I'm showing here is just the beam trawl sampling, and there is some. Yeah, so I'm not showing the surface trawl data, but that's the pattern that we saw, is that we would see consistent numbers of saffron cod, and then the warm temperature came and the saffron cod disappeared. They were gone. But what we see here, and this is kind of the signal, is really emphasizing the signal, is like, it wasn't that they responded to this cold temperature, and as soon as it started to cool down, we're seeing the presence of these saffron cods. So I think that the timing and the lag of how these populations are responding to temperature, I think, is important for understanding kind of what's the underlying mechanism that's causing catches to vary within the survey area.
So have you routinely seen the young of year peacock in the surface trawl?
It's that's a different story. We haven't really seen many young of the year peacock in the Northern Bering Sea, and it's only been recently when we started to see young of the year Pacific cod become abundant in the Northern Bering Sea.
Okay, that's what I was wondering. Yeah, so you don't really know how unusual 2020. Well, so I guess the 2023 is really perhaps, well, you only have 3 years, but that it might be higher now than it used to be, based on your longer time series, or is it too small a sample size that you can't really tell?
Yeah, I wouldn't be surprised that what we're seeing new spawning aggregations of cod, and so now we're actually seeing a lot more cod in the Northern Bering Sea than we did historically.
Yeah.
So there's a signal there, but yeah, I'd have to show the surface trawl data to really make that clear.
So you're talking there's more. Oh, sorry, Franz, go ahead. Didn't mean to jump in.
I was going to ask a different one since I have the floor, but go ahead.
Okay.
Well, if you want to follow up on that, go ahead.
No, go ahead. Seriously, Franz, go ahead.
Okay. Well, the other one was that I was curious about, and I'm not sure if I completely followed that. I think you said that you showed the ichthyofaunus and the pollock, and you said that that's a likely source of ichthyofaunus for Chinook, and I was wondering what that's based on, because the Chinook have had ichthyofaunus for for a while, right?
Yeah, and I think this is just an idea. I think we need to know. I think my point is that we need to kind of understand what the forage base is for Chinook and how that's changing. Because if with the collapse of herring and capelin in the Northern Bering Sea or the Eastern Bering Sea shelf in general, at least for the size ranges that we sample, that means that the diet of Chinook salmon is going to diversify, and they're going to start feeding on other prey species that they typically don't feed on. So I think in pollock, I think they're the reservoir for Ichthyophonus. So whatever conditions that will cause Chinook salmon to feed more on pollock than they have historically is likely to be a source for why Ichthyophonus would vary. I mean, it's a concept. It's understanding that it's there. It's age one. They don't have to eat offal. It's there in the age one population. If they feed on age one pollock, they're likely to contract Ichthyophonus.
And that's kind of known that they generally get Ichthyophonus through prey.
Yes, they get it through their food. That's the only way that they get it. They will eat an infected prey species. So unlike in the Gulf of Alaska, herring are the reservoir for Ichthyophonus. But in the Bering Sea, there is no Ichthyophonus in herring. Rather, pollock serve as that reservoir for Ichthyophonus.
Okay, thanks. I'll let others ask questions.
All right. So thank you both. And we did have a caller, and boy, howdy, I'm sorry I didn't call on them. So Doug Molyneux, go ahead, followed by OA.
Great talk. If you go back to that benthic ecology slide, I think it was slide. Yeah, there we go. I'm just curious on the juvenile snow crab, how common is it for one age class to dominate the abundance of snow crab? You see that sometimes in herring. I know one age class will kind of march through several years, and then you'll have dry periods, afterwards. So any ideas on that?
Yeah, I'm not as familiar with the recruitment dynamics with crab. I know that it's challenging for snow crab because I think it's a bit unclear what the ages are. So all that you can kind of do is you look at size distributions. But here I think you can see the progression of that from 2021, where we got a lot of these small crab. So we're like, oh, this is great. This is what we're going to do. And then, since then we haven't hardly caught any of these really small snow crab, less than 15 millimeter carapace width.
Yeah, so that's really 1 year class marching through those three.
I think so. That's kind of what our sense is. But again, so we're able to see this now. It gets really confusing in a hurry, and I'm not an expert in snow crab, but at least I think we have the potential, I think, to capture recruitment events in snow crab with this small mesh beam trawl.
Yeah, definitely. And I guess for context for folks, this is in millimeters for carapace width. The legal or the preferred size for commercial is I think it's 140 millimeters across. So those crabs have a long way to go before they reach 140 millimeter carapace width.
Yeah, it's going to be a while. Yeah,
several years. Yeah, thanks. Great talk.
Well, thank you. Yeah.
All right. Thanks, Doug. Thanks, Jim. Okay, OA, go for it.
And OA is me, Gay. I'm using my cell phone as a mic and my computer as the screen because it has a bigger screen. I'm looking at your benthic ecology screen by Dan Cooper. And I also had questions on the snow crab too, but so Doug already raised that question. That was my question as well, pretty close. So I was thinking with that, excuse me, the ocean seafloor, I know there's been so much changes under there, for the crab and the crab and the fishes, and then what other fish species or invertebrates under there, the changes under the ocean seafloor. We've had so many earthquakes And then I just saw a map earlier. There was, I didn't know there was an earthquake not far from Diomede this year.
Hmm.
They probably have them a lot, yet we don't know. I mean, yeah. So I'm thinking with those, with the earthquakes and the changing of the ocean, the changing of the ocean temperatures. Because when I think back to the first time, and I know Gay has probably heard me say this a number of times, but I'm going to say it anyways, that in 2016 we saw sand on our beach and everybody freaked out. This is little Diomede, we're all rocky, and then there's sand all of a sudden. Then we're having weird species of fish and invertebrates we've never seen before, ever. Not a traditional harvest for us that we ever, were a part of, but they're coming ashore and we're finding them, So I'm wondering, since these changes, and I know there's changes, I know our prevailing winds have moved from the south in the fall season, when we usually have the north wind, that carries the cooler temperatures and, that kind of thing. I know that's changed. But then, so looking at your benthic ecology slide and the snow crab condition studies, that I'm very interested in that one. That's what I want to learn about. From this, if we could have maybe Dan Cooper, maybe shed us some light on the snow crab condition studies, that would be a good one. The other one I was looking at was the ice algae food web effects, your sediment samples. How are you collecting your ice algae samples?
So you're hitting all the points that is the kind of the broader ecological studies with this sampling program. And so a lot of what they're trying to do is they can track I think ice algae through the fatty acid profile that is present in the food web. So a big part of what they're trying to do is look at the fatty acids of these snow crab and be able to identify what the source of the carbon is. Is it phytoplankton or is it ice algae? And so that work's all ongoing, and I think they hopefully will be ready to present the results of that work, and that given a straight science presentation focused on that question instead of this big broad sweep of everything that we do on the survey. I think that would be good, and those folks can tell you much better than I can about the results, but yeah, it'll come soon.
So for the harmful algae, Opic, I'll add that I believe the surface trawl survey, are you providing samples to the EcoHab project in the Northwest Fisheries Science Center? Are you
Yeah, we've been providing samples to Kathy LaFay for probably 6 years. Yep. So she's been monitoring harmful algal toxins within the whole gamut of benthos to fish and also just the phytoplankton samples. So she's monitoring that every year. Yeah.
So if that helps, Opic, too, that they're sampling to get at the harmful algal situation as well. Thank you. Go ahead. Keep going.
And yep, she's also taking my samples as well. So with the ice algae, I'd like to learn more about the ice algae. Oh, well, I'm thinking your ice algae food web is just looking at the food web for the fish. I mean, what fish are you eating?
Yeah, I think the focus of that is tied to snow crab. But it would also apply to fish. But I think their particular focus is looking at that connection between recruitment events in snow crab and their dependence on ice algae.
So are you taking samples or tissue from the viscus of the crab?
Yep. All the crab are saved and frozen at -80, and so they do the whole spectrum of fatty acid analysis on these small crab.
Cool. Okay, that's awesome. That's cool. Okay, those were my only questions, Gay. And
Rock on.
thank
Thank you, Opik.
you, Jim.
Go ahead, Dean. You're up at bat.
Yeah, I had another question, and this one's about Ichthyophonus.
Mhm.
If you move into the Atlantic, if you move into the Baltic Sea, the main source of Ichthyophonus comes through herring in that ecosystem. And I know when you're over in the Bering Sea, when the Chinook come through, whatever they're grazing on, and they pick up the Ichthyophonus, it doesn't start manifesting until they start migrating up the Yukon. With all the stress that they're going with saltwater to freshwater, that's where they impact. And there's a group of fisheries people studying Ichthyophonus on the Yukon. And do you guys engage with them saying, oh, we had a low herring season and a high this capelin season, and maybe the capelin are bringing it some food source for the Chinook. I was just wondering if there's a connection there that you're trying to make, because there's a lot of work done on both sides, and it's all good work, but you still need the communications.
Yeah, that's a good point. Yeah, all the scientists that are doing the work in the Yukon are actually, like J.D. Ferguson is leading all the Ichthyophonus research in the Yukon River. So this work we're doing here is just to add context to the research that they're doing in the Yukon River. One of the goals that I would like to see folks, the JD and other folks do is start to understand kind of that process and the mechanisms for Ichthyophonus, and potentially be able to provide a leading indicators, like, is this going to be a high Ichthyophonus year or so I think with these marine surveys, we may potentially have the ability to provide the managers and stakeholders as to what to expect for Ichthyophonus.
Thank you. Appreciate that.
Yeah.
All right. And before we get to Alex, I just wanted to say, for everybody who's from the region, feel free to, if you've got questions in the Bering Strait region, specifically on this talk, feel free. You can jump in. We lost the caller, but actually it was a New York City caller. I had to look up while you guys were talking. I looked up the area code. It was kind of a shocker. But anyway, feel free, if you're uncomfortable speaking, then type up something in the chat and we'll get your questions answered, because the audience is these presentations are for the Bering Strait region. And I know I've got some questions, but I have to be polite and wait to the end. So with that, Alex, go ahead. You're up at bat.
Yeah, can you hear me okay?
You bet. Mm-hmm.
Well, thanks, Jim. Excellent presentation, and thanks to you and your colleagues for getting out there and rocking and rolling and keeping these important data series going. I got a multi-part question on your forecast. How is the forecast shaping up for 2024 for the Yukon Chinook, and are you making a forecast for chums, and are you working towards one for pinks now that you've got the pink salmon CPU data coming in?
Yeah, so it gets a little bit complicated. I think for 2024, there's, this figure here kind of shows what the forecast would be for 2024 based on this juvenile model. The actual forecast that is provided for this stock group is part of a U.S.-Canada Joint Technical Committee group, and so this is just one of the models that are considered in the actual forecast for the Yukon River. And so this is one piece of it. The outlook for the Yukon is a blend of a dynamic Ricker, dynamic sibling, and then the juvenile model. And we may potentially, move into, I'm thinking have to move into a dynamic juvenile model, just because we're just not seeing the forecast, the runs come in. It's just consistently coming in below our forecast. So until we can find out why or have a correction, then we may have to move those to dynamic models. So the chum salmon work, this is work that's being led by Sabrina. She's working through a number of things. She's trying to build spatial models for juvenile abundance. So she's going down a route that's different from this simple kind of design-based estimators of abundance. My guess is hopefully we'll see something within a year. But it's a lot of it's her schedule and progression of that. So kind of really dependent on her. And we did develop a forecast model for pink salmon. Excuse me. And we, following the excellent work that you did in Southeast Alaska, we figured that we could probably use the pink salmon catch rates at least in the Northern Bering Sea to project what the run sizes would be for the Northern Bering. And doing so using log models and kind of dealing with the patchiness that occurs in pink salmon in particular, we were able to show that there is a reasonably good relationship between juvenile and adult returns. But without pink salmon are not as important from a subsistence perspective. So they've kind of been not at the forefront of what we're doing here, but I think Ben Gray at ADF&G is starting to take those pink salmon models and we'll probably dust that off. Ed Farley published the original pink salmon models. And yeah, I don't know if I hit all the questions, but
yeah,
did he answer
great.
your? Okay,
Yeah.
you're good, Alex, huh? Okay. Thanks, Jim. And Doug, followed by Wes. Go
Yeah,
ahead, Doug.
another question there on, I think it's on slide 9. You mentioned about the high energy content that you saw
Yes.
this year in chum salmon and pink salmon. And maybe it's too early, but do you have any genetics on that? Do you know the stock of origin of those chums and pinks?
Well, pink salmon are tricky because you can't they're all kind of one big group. You can identify like Bering Sea and northern Russia pinks that can separate out from, say, Gulf of Alaska, but that's all one largely homogeneous group. It's for chum salmon. So the index that Sabrina's working on is just on the fall run, which is the upper Yukon run, and they're genetically distinct from all the other chum salmon stocks, but they only make up about historically they've only made up about 20% of the chums. So obviously more recently it's probably a lot less than 20. So maybe somewhere in the range between 10 to 20% of the chums are actually fall chums. So this is more of it's a combination of both summer and fall chum salmon. It's an assessment of all the chum salmon, but the chum salmon are largely we avoid sampling or using the I'm trying to remember if we have those. Kotzebue Sound chum salmon will come through the Bering Strait, and that can have an effect. So what we've done is we've limited the spatial extent of the chum salmon that we would index to try to just target the locations in the survey where we expect the Yukon or Norton Sound and Yukon stocks to be. So it's more of a Northern Bering signature.
Okay, thanks. I have one more question I think might be kind of quick, so I'll
Yeah.
warn it in here. But on slide 14 about the low, let me see. Yeah, with the Canadian origin Yukon fish coming in consistently way lower than the forecast, more so than the overall total Yukon tongue type. To what degree might ichthyophonus be a factor with that Yukon or Canadian origin fish being exceptionally low below forecast?
Yeah, I think ichthyophonus is definitely having an effect on that, but not just ichthyophonus, but heat stress, thiamine deficiency. They are coming in deficient in egg thiamine, which has just a whole host of other things that will cause problems. And so there's probably multiple factors that are contributing to this kind of stock difference in our ability to forecast runs. So I think there's more work that's needed, and I think if we can't figure out exactly how to correct this, then we'll have to go back to a dynamic model when providing projections.
Thanks.
Yeah.
Fantastic presentation. I appreciate you taking your time to present this information to us, and for Gabe.
Well,
So thank
well,
you.
thanks, Doug. Yeah. Appreciate your questions.
All right. Wes, you've been so
Okay.
patient.
Yeah, no
to see you.
problem. Thanks so much, Jim, for giving the presentation, and it's very helpful. I did want to make one point. Your comment that pinks weren't as important. That may be true on the Yukon and the Kuskokwim, but here in Norton Sound, pinks are, I think, have become more important, or more important than chums. And one of the reasons for subsistence, and one of the reasons 2023 was so hard, much harder, I think, for most people compared to 22 and 21 in Norton Sound, was because of the lack of pink salmon.
Oh, wow. Yeah. Fair point. Sorry. Yeah.
Yeah, no, I just to note, I think I just wanted to note that in the importance of, each region's different, and here, and how, when I first came to Norton Sound, I was amazed at how important pinks were here compared to other places in Alaska I'd been. So
Well,
thanks.
Yeah.
yes.
Well, yeah, thank you, Wes. And I think that's a really good point. And I think we need to carry pink salmon through these AKSSF objectives, and not just get too wrapped around chum and Chinook. So that's a good point. And I tend to be very Yukon centric in my experience with salmon in the Northern Bering.
No, yeah, thanks. And that's a very common thing that we run into here, is that, yeah, the Yukon and the Cusco with their Chinook are, and we do have Chinook issues here in Norton Sound also, but they seem to override sometimes of the other species. But thanks for bringing it, and I really appreciate seeing the information on pinks that you've provided. So thank you.
Yes, we'll make sure that it gets into our proposal process for this next round.
All right. Well, I've got some questions if you're still game, Jim.
Yeah.
All
I
right.
got no place to go.
Okay, so here's what I heard, and I'm sort of more thinking of the region. I'm not a salmon aficionado. I mean, I like to eat them, and they're wonderful, but I don't study salmon. So what I heard overall was that large, greasy copepods, the zooplankton, have gone down in number. And we haven't heard anything yet about ice algae as far as amount. But, we've got a lot less ice. We have a lot less ice algae being made because the ice isn't stable enough and around long enough to make those big mats of it that used to be underneath, I think. And then just the way it doesn't extend down into the Southern Bering Sea, it's got to be less ice algae. So then I see, okay, less of the copepods, the little, not a kingock exactly, but a copepod. And so, the birds eat those, the small fish eat those, the bowhead whale eats those, jellyfish eat those. And then we see that there's no juvenile snow crab. So those numbers have gone down. And then the forage fish is terrifying. That seems to be going down. Is this, but you've got, just letting someone in the waiting room. But you've also got saffron cod returning, Pacific cod doing well. We've got some issue. I see that with the salmon forecast. Is it, at what point, what data does NOAA need to say at an ecosystem level for the Northern Bering Sea that the carrying capacity, the ability of our ocean to feed all our guests, has that changed so that we've seen such hunger in things like our seabirds for 7 years in a row, multi-species seabird die-offs, since 2017 when the big, we didn't have a lot of ice. The thermal barrier wasn't renewed between the southern and northern Bering Sea. And then all these things just came racing north because the water is rolling north in our region. And then we've seen our first UME on starvation, mass mortality due to poor body condition in 3 species of seals that want juvenile seals, typically, that want small forage fish. Is there, I mean, is there something you can say towards carrying capacity? Can we say anything about carrying capacity?
Yeah, carrying capacity is going to be really difficult to get a handle on, but there are things related to carrying capacity that are currently being studied right now. So like one of the things that Todd Miller is looking at is he's trying to look at what the trophic overlap of these large gadded fish and other species like herring and salmon. So once you get an idea of how much they're actually overlapping, you will be able to see what the potential is for competition. But until you've established how much actually there's diet overlap, then it's kind of hard to even determine if there's competition, let alone carrying capacity.
Well, I'm thinking, yeah, it's a good answer, and I appreciate that. I'm thinking you're talking about fish. I'm talking about even going further back. Are we making enough algae to feed enough krill to make the fish satisfy the fish and the birds and the bowhead? I mean, it seems like everyone's gotten in the Northern Bering Sea, and from my just as a resident here, we see hunger in the Northern Bering Sea, Bering Strait region, where we shouldn't see hunger in the Northern Bering Sea, Bering Strait region. And is that a function of overall at the beginning of the food chain, at the algae and then the little krills, if they're less, of course, then we still have all the big guests, all your fish, your marine mammals and birds and crab and all these things, but they're all hungry. Are you guys looking at that bigger picture of, has there been a food reduction at the primary level?
I think so what I think we're gonna see here in the next year or two is the North Pacific Research Board is gonna launch their integrated ecosystem research plan, which is going to be basically building models, studying the whole integrated ecosystem. So I think once the proposals and the research come through to look at it from that perspective, I think we'll have better answers as to what's driving it. Because a lot of these things are going to be based on models. We'll have some observations that can help constrain and develop parameters, but a lot of those questions are going to be kind of a model product.
And then my second question, I got a couple, so bear with me. My second question is, and I think it was Doug Molyneux actually, or somebody, maybe that Alex asked it, and that was, given what you know, we're headed, currently our ice is not so good. And our temperatures, we did get a cold spell, a cold snap, but this is middle of February. We're getting in on the middle of February, and we have open water. We're going to have a rain event here. They're warning us tonight and tomorrow. We're having a 5 to 7 inches of snow coming out of the southeast, southeast for Nome. It's going to be pumped up again, and we're unseasonable. And if it just stays, we're in February, March, April will be here before you know it. So, do you have any, if it's bad news, we'd like to hear it so we can be prepared. If it's good news, we'd like to hear it because we'd love some good news. What's the forecast for some of these salmon? We know they've been coming in under the forecast, but is there some light you can shed on that? A best guess for 2024? Should we be optimistic or?
No, I think all the juvenile data for Chinook is indicating that things are probably not going to get much better. One of the things that could change, however, is with not just marine but freshwater temperatures starting to cool. Hopefully, they'll have less issues during their spawning migration. So there could be some improvement there. For chum salmon, I would say, although our big concern is just the nutritional status of the chum, and this is not just in the North Bering, it's also in the central North Pacific, but seeing the chum salmon kind of being at record high levels of lipid and in condition. I think
So fatty.
yeah, they're in really good condition this last year, but
Okay.
you know, their abundance is not. It's still way below average. So they may improve, their survival will improve, but I think we might be seeing some evidence of that chum salmon numbers will hopefully start to improve, but it could be a while before they'll get back up to the level where people can start harvesting them for subsistence.
And just so you know, we didn't talk about transboundary over to Chukotka, but their salmon aren't doing very well. Do you get any news from there?
No, and I think especially after with the Ukraine situation, there's been really the opportunities to work together with the Russians have largely disappeared. So
Well, you should tune in with Gamble and Sivunga and Diomede and Wales. I mean, when you're in the Strait, the people are fishing closer to Chukotka than Nome. So
yeah.
I'm just saying that might be something to think about. And then my
Yeah.
last question, and we really appreciate all your questions, and this shows my ignorance, but you had mentioned, okay, so the chum are fatty and but their numbers are way down, but Chinook salmon, thiamine ace. So you said they're eating something different, perhaps, that's giving them that they're eating that has thiaminase,
Yeah.
and so it's breaking down their thiamine. What is thiamine? And that might be a problem for your Chinook, right? You're looking for why the numbers are so down. So can you tell me, what does thiamine do for a Chinook salmon? I don't know. I have no idea. No
So
more idea.
thiamine is vitamin B. So it's
Yeah.
absolutely critical for like the Krebs cycle. So it's like if you don't have thiamine, you're not going to be able to metabolize food or anything. It's just like they will suffer, but there's like lethal levels. Like so the difference is like the female they can tolerate low thiamine. They can then where the critical period in low thiamine levels in salmon or in any animal is during the embryonic stage. So it's during the embryonic development stage. It's just like if the fetus doesn't have the right nutrients, they're gonna suffer from that. So that's where we've been able to show that there actually are lethal levels of thiamine, such low levels in egg thiamine and Chinook that there's no reason to believe that any of the fry will survive from that female.
And none of the other species have this deficiency.
Well, so the deficiency results from feeding on fish. So fish that have thiaminase. So because Chinook salmon is so piscivorous, they feed heavily on fish. They're the ones that are going to be most susceptible to thiamine deficiency. And it's caused widespread mortalities through, like, particularly in California. They've been able to really clearly link it to just widespread mortality of Chinook salmon in California.
I guess, why now? If they've been eating fish all their livelong days, why now? And if it's
it's a good question. It's like, what's changed? Well, first of all, we know that the forage community is completely changing. So just by the fact that they could be feeding on different prey that they normally don't, which would cause it. We also show that thiaminase activity levels can vary quite a bit from year to year. So the toxicity of, like, say, rainbow smelt can be very different under certain situations, and other ecological or environmental situation, that toxicity level could be much higher. So there is that shift in the level of thiaminase activities within these prey species can vary.
All right. Well, maybe that's something they can check with the birds, because the seabirds might be shifting, and so maybe they've got thiaminase.
Yeah. So at least this point, we've only been able to detect thiaminase in rainbow smelt, which is a good thing. It just means that, hopefully they have a diet that isn't just rainbow smelt. Otherwise, they can run into problems.
All right. Well, thank you, Jim. And we've got Dean Stockwell. Go ahead, Dean. Bring us home.
I hate banging on you. You've done a great job. So be patient with me, please.
Yeah.
So as a suggestion, you collect copepods. Why not also test them for ichthyophonus? Because they can be transmitters. And whether it's euphausids or copepods as a whole, think about that as an addition to your study. And I'd asked earlier in chat, what fish species do you actually test for ichthyophonus?
So
and if you can't answer that right now, send a message to Gay. She'll get it to me.
no, this table shows all the testing that we've done so far. So we've tested herring, rainbow smelt, sand lance, capelin, pollock, age 0 pollock, and then age 1 or age 1 plus pollock.
Any of the other forage fish?
Well, there's not many other forage fish that Chinook salmon feed on.
Okay, just curious.
Yeah,
You did
so
a
we
great
try to
job.
we try to get all the species that we know Chinook salmon feed on. But there
thank
could
you
be a few others.
Thank you for your patience.
Yeah, no, thanks, Dean. I know it's through asking questions that I, find where I'm kind of missing content or I skip over things that aren't as clear as they should be. So appreciate the questions.
If you come at the Fairbanks, we could have a conversation. Anyways.
Yeah, we'll talk more about it. Yeah, you're welcome.
Opic's got her hand up. So thanks, Dean. Go ahead, Opic.
Yep, thank you. And thank you, Dean. And because of Dean's question, I thought of one as well. Like, so for the bottom fishes, like the sculpin fish, which used to be very abundant here in Diomede. That's our traditional food source right there. And we used to be able to catch it anywhere, pretty much out here in front of us, either from on the north side, in the middle of the island, and then south side. You can catch the sculpin anywhere in the winter season, when we usually catch them, winter and spring. But then it's been quite a few years now. Excuse me. Quite a few years now, we are having such a hard time finding them. Everywhere we try over the ice to drop our lines to catch them, we're not catching them. So we're wondering, where have the sculpin gone? And then we used to get the Arctic cod, the blue cods. We used to get those too every spring season. We can catch those, but now we cannot find them. A couple of years ago, we found some. What year was that, Gay? Maybe 2017. We found there was a bunch of them, but then now where are they again? We're not finding our fish. So
thanks for bringing that up.
Yep. So I'm just wondering if in the near future somewhere you do your trawling, maybe test those bottom fishes too.
Yeah, so the bottom trawl survey does those sculpin and the Arctic cod actually are going to be bottom associated, so they're not going to be up in the water column. Okay, so the best information that you're going to get on the abundance and distribution of those species are going to be from a bottom trawl. I mean, our trawls, we avoid coming into contact with the bottom with our rope trawl, and so we strictly kind of work on what's happening up in the water column. So that's where herring, salmon, sand lance, capelin, those species are going to be up in that water column. Those are the species. But we do. I think in general there is a bigger concern over Arctic cod throughout the Arctic because they're like seabirds and a lot of other marine mammals are extremely dependent on Arctic cod. And so they're like the main kind of trophic transfer, like everything kind of from the lower trophic levels up to the upper trophic levels happens through Arctic cod in the Arctic. And so seeing those populations decline has widespread impacts to not just the ecosystem, but as you're saying too, to communities in that region. So
okay, so your trawls are mid trawls?
Yeah, so we sample like typically I sample the upper 20 meters, so upper 60 feet, I guess that would be. So that's the layer that I sample in our trawls. And so that's about half, probably the water column, the upper half of the water column, and the lower half is not sampled in these surveys. Except for this small mesh beam trawl. Small mesh beam trawl, it's tiny. I don't know if So it's not big enough to capture. You can kind of, I don't know if you can see this, it's a very small trawl. I mean, it's 3 meters across and like 1 meter in height. And it's very small mesh. It's designed to catch these really small crab and these tiny, they're very small benthic fish.
All right, and I put in, this is Gay, I put in the link, I mean in the chat, the link to, I think it was Dwayne Stevenson's 2023 bottom trawl survey results, and they do, if that helps OPIC, they do get at sculpin. So if you want to watch that, or let me know if you want to contact Dwayne, we can set something up. He can call and talk more about the sculpin, because that is a huge, bullheads. That's hugely, wildly important for the Bering Strait region, especially in the Strait. So, I mean, that's a good food source. And a lot of times people, it's always salmon, but everybody's got a little bit something different in their backyard, and in the Strait, you know, why not bullhead?
Yeah, and I think that if making that connection to subsistence, traditional fishing, food fishery for these species is really important for these people to understand and recognize that that all of a sudden now that really elevates the importance of that particular species. So it's, I think, yeah, definitely connecting with Lyle, Britt, and Dwayne, those guys.
Let me know, and I can we can set something up.
Yep, I'm for it. But yeah, we'll set something up. That would be great. Thank you. Thank you again.
Well, thank you. Yep.
All right, I do have one last one, if that's okay, and it's slide 15.
Yep.
It's slide 15.
Yeah. Go ahead.
Just to be fussy, since you're so nice, if you can go to slide 15.
Yes. There we are.
There you are. Okay, so this is fall Yukon River chum salmon abundance.
Yes.
What is the, and these are from a model where you're just running your best guesses, and this, they put all the data in, do the math, and this is out, or is this the actual fall Yukon chum salmon abundance?
So for the entire time series, except for 2023, because we don't have genetics yet for that.
Yeah.
All the other years, what we have is kind of an overall chum salmon abundance for the Northern Bering Sea. And then we have a stock composition for fall chum. So if we know that 20% of that year of the chum salmon were fall chum salmon, then the total index gets multiplied by 20%. If the next year it's 5% fall chum salmon, then that total index gets multiplied by the composite.
So CPUE, for people who may not know what that means, that's catch per unit effort. So I'm taking a stab here, but if the circle's big, you're catching more fish with less effort.
Yeah. That just shows you the distribution. Here you can see there's two distributions. There's like this group of chum salmon up north around the Bering Strait. That was a little unusual for us to see that. And then there's this bigger group further south. So one thing we do need to test for 2023 in particular is to find out if these are Kotzebue chum salmon that are coming down the strait, or if in fact the Yukon chum bifurcated with portions of them going north and portions of them going south. But we'll be able to figure that out once we do the genetics.
So my question is, what on the left does the black dotted line represent?
Oh, that's the average.
Of all those from 2000,
every
the
year.
last 20 years.
So if it's above the line, it's above average. If it's below the line, it's below average.
And it's catch per unit effort.
Yeah, so this is just a density
juvenile
index. It's not an actual total abundance index. It's a density index. Yeah.
So it means that people have been trying to catch, and the return on that effort is less. So they're catching less fish. That's how you'd read that?
Well, it just means that if we took those circles and you just averaged
on the left,
whole survey, you would come
bars.
up with the density estimate, and that's what that is. It's the average density of chum salmon.
The bars on the left is what we're talking about. Is that correct?
Yeah.
Okay.
Yeah. So just with the
number
number of the number of fall chum salmon per kilometer swept with the trawl. That's what that index is.
Okay. All right. Well, thank you so much. Any other questions for Jim? Jim, I want to thank you're over the mark, and we're very appreciative calling in from Juneau. One more if you can take it. Go, Opic.
Okay, Opic, what you
Okay.
got?
Sorry about that, Jim. Just one
No,
more.
He's got to have dinner at some
Super
point.
very important one, and I think this goes for everybody too. So you know these Alaska saltwater fisheries books, the one Kessler did in '85. I know there's an updated one. Gay gave me a book, saltwater fisheries book, but how often do they update those books? Yeah, especially for now, because now we're seeing so many different species of fish. You know, some are invasive, some are not. But you know, it would be I would like to know when and how do they get these updated, you know, like for the Bering Strait.
Excellent.
Like what type of species are in our waters, invertebrates and fishes, you know, what's in there, what's in there now.
Yeah.
You know?
Yeah, I think, you know, Kitty Mecklenburg did an atlas of the Arctic fishes, and so that probably is much more, it's updated, so you're going to have more spectrum of species in there. But as this region continues to warm, the species in this region and in the Arctic are going to change.
Opic, it's an excellent question, and we've got some leads on and some ideas on that, and have been talking with Kathy LaFay, and it needs to get done. Because my last question, Jim, I guess we're going to have a stringer here. The last question is, how do we all, we're on the coast, like Opic says, where are these guidebooks to help us? How do we help you? What do you need from us?
When you find a fish,
to hear about
when you find
help us.
Yeah, when you find a fish that you've never seen before, you have no idea what it is, save it. You can take a picture of it, but the more important thing besides just taking a picture is save it, because it's really oftentimes it can be difficult to have a confirmed identification of a fish just from a photo. So save it, send it here to the lab, and we'll tell you what species it is.
What do you even
Save it now.
So send that to me and Noam, I would imagine.
Yeah, send it to Gay. Yeah, because then, just if you see anything unusual, something different, save it, send it to Gay.
Well, I think Jim, that Dwayne and Lyle Britt with NOAA Race Division already have the corner on that market. They are already identifying. We send them all manner of fish photos and things like that. But you also, there's nothing you need from us other than an unusual fish. But if we're seeing, what does ichthyophonus look like? Is that something you brought it up in this thing? Is that something we need to be like eyes out for that? Or is that something that it just sort of kills the fish?
So go ahead. Is there a comment? I can tell you that the progression of disease usually occurs within freshwater. And so it's when they're in their spawning migration, you can identify ichthyophonus if you see like white spots on the heart. And then
so it's not an something we would see on the coast.
You cannot necessarily tell from looking on the outside of it.
Shucks. All right. Well, if that's so, it sounds like if you see something, report it. I'm Jim Murphy's in Juneau with the Alaska Fisheries Science Center. I'm here. I'll always take reports and get them to NOAA personnel if they.
No, but I think it's good. I think
you guys turn it around and we
turn
it
with
around.
You, and then from there, it's just recording these things, and
we're doing that.
Anything that's unusual, document it.
Yep. And I do for Gay. I do this for Gay Sheffield every year, all the time.
Good.
Isn't that right, Gay?
Yeah. And actually OPIC really does. And even if, even if it's healthy and you just want to know what the name is, so we can look it up in a dictionary or like a guidebook, like OPIC's talking about, learn more about this kind of thing. Even that, that's why we don't have really good guidebooks for the Northern Bering Sea. And it's more than frustrating, especially nowadays when there's so many
new species.
Yeah, new species.
Yeah, like why are these warmer water fishes up here?
And marine mammals and birds.
Yeah. And yeah, I would say too, even, I mean, it can apply to invertebrates. It could, you know, like, oh, there's this big ball of goo that showed up on the beach. We have never seen it before. Well, you know, take pictures, save it, send it to Gay.
Yeah, do. Seriously, you know, I'll try to get an answer. I'm a
Yeah,
public
but
servant
it's,
of the region. Oh,
yeah.
geez. Okay, Doug, after you, that's it. We
Okay,
gotta let
Doug,
this
jump
man
in
go
there.
home. We gotta let this man go home. Go
Just
ahead, Doug.
real quickly, rather than taking just pictures and sending a whole fish, I mean, how about for genetic samples, just a tissue sample that can go along with some pictures? Would
Doug,
that-
now you're getting into what
Yeah.
everybody, no, I appreciate that. I appreciate your comment. Yep, even a little, a piece of it, I guess, as well as a photo, right? That's what you're asking.
Yeah, and a lot of things can be reasonably identified with a photo, but a lot of times if you just take pictures and you don't, you know, you don't know whether the picture is going to work, and sometimes it's like people won't officially identify that as being recovered there unless an ichthyologist looks at it and says, yes, this is that fish. So ichthyologists won't just take anecdotal
Well,
reports.
the bird people want birds, and the marine mammal people want marine mammals, and the ice people want ice reports. And I mean, that's where it starts to snowball, and that's why it gets.
Well, and it's just that
I'm
that's
trying to
where
think of a patient, kind word to say about that, but we need the help. We need to know these things. That's why I'm asking. We need the urban-based researchers' help in understanding what's going on. But at the same time, people are volunteering their time. This is a food security, food safety issue. So yes, we will take pictures and tissue, and we will do what we can. But at the same time, boy, howdy, we would sure like. And Jim, that's why Strait Science, you know, you came here tonight and gave us a little bit of what happened during the surface trawl survey. It is really important for us to hear this news about the environment and the fish and the forecast that you've got, even if it's not good news. We need to know, because people have to keep feeding their families. So we appreciate
Yeah.
it. We're not shy to hear bad news or good news. We just want to hear the news, and we will do what we can, Doug, to get you tissue bits and sightings for Jim. But know that we really need to be working lockstep more, and that's a personal thing. But anyway, thank you all, everybody, for coming to Strait Science. And the next Strait Science speaker was in the house tonight, but he's left to finish his dinner, and that is Franz Muter. Franz Muter will be giving us a talk on the 22nd of February, and he's going to give us, it's not really results so much as a discussion about subarctic and Arctic fisheries. Thinking about what does that mean for everyone, both on the coastline to at sea, to transboundary issues with that, to potential conflicts, to potential benefits. And it's going to be a good conversation, I hope. And Franz is the one that can handle that, because he's been talking about subarctic, that's us in the Bering Strait, and Arctic fisheries, which would mean north in the fisheries world of NOAA. That means north of Diomede, like the north end of Diomede north. And so what I think Franz is going to do a great job, and he wants to have a discussion, just to even start a discussion about good, bad, or ugly. And like I say, Bering Strait is not afraid to have a good conversation, because we're living it. And so I'm sorry Franz got off. He must have had his dinner ready to eat. And I look forward to that. So tune in. There'll be notices on that for February 22. And with that all, thank you so much for hanging in.